Hi CSC students, and welcome to the Introduction to Cells and Microbiology for your NSCI3785 course, which is Introduction to Companion Animal Biology. We're going to have a quick look and see what's happening here, bit of an intro, maybe some brainstorming opportunities for you. We're going to talk about cells, we're going to focus a bit on bacteria, and then look at eukaryotic cells, which are animal cells, and how they're organised. The lab that goes with this particular session is the first one in our actual course, in the block course, so you'll see that when we get there. A couple of things that you could think about: what is a cell? What is a cell membrane? And what is the nucleus? And how would you find it? And how would you find that? What would you find in the nucleus? Let's have a look at this video.
There are many kinds of animal and plant cells that have different architectures and do different things. But many of the parts of these cells are the same and have the same functions. If we created a typical animal cell, it might look something like this. Even though no specific animal cell looks exactly like this one, it contains features shared by most such cells. Surrounding the outside of the cell is the cell membrane. This membrane is very thin, only about 1/100,000th of a millimeter thick. A photograph of a cell membrane taken with the help of an electron microscope reveals that it has 3 layers, 2 dark outer layers sandwiching a clear middle layer. The cell membrane acts as a boundary between a cell and the environment outside, but it also allows certain substances to pass in and out. Particles of food, for example, can enter through the membrane so that they can be used as raw materials for manufacturing and as fuel for energy. Oxygen can also enter a cell to be used for burning fuel. Other things pass out of the cell through the membrane, including waste like carbon dioxide gas and important chemical products that the cell has manufactured.
Inside the cell membrane, there are many different structures called organelles, which literally means little organs. One organelle, the nucleus, is the control center of the cell. The nucleus has a membrane that has pores in it. These allow certain molecules to enter or leave the nucleus. Inside the nucleus, there is fiber-like material called chromosomes. Chromosomes contain the cell's genetic information. This genetic information is like an architect's blueprint that determines how wood, glass, and other materials are used to build a house. The genetic blueprint in a person's chromosomes determines how different proteins are put together. These proteins are responsible for physical features such as the color of a person's hair and eyes, the shape of her nose, and even whether she is susceptible to certain kinds of diseases.
From the information contained in the chromosome blueprints, the nucleus sends messages to the cell's cytoplasm, which makes up everything else that is inside the cell. The cytoplasm is composed of water, minerals, vitamins, salts, a variety of chemicals, as well as different organelles. Some of the organelles are called endoplasmic reticulum. Among other things, endoplasmic reticulum serve as the cell's assembly lines because they are where protein products are put together based on instructions from the nucleus. The endoplasmic reticulum serve other functions as well. They're like highways that provide a transportation network. Among other things, they connect different organelles in a cell and allow them to exchange materials.
Mitochondria are another kind of organelle in the cytoplasm. Inside a mitochondrion, there are highly folded membranes. Here, chemical reactions take place that use oxygen to burn particles of food for energy. Mitochondria are often called the powerhouses of the cell because they are responsible for providing for the cell's energy needs. The number of mitochondria varies in different kinds of cells. Very active cells, like muscle cells, contain thousands of mitochondria.
Before particles of food can be burned in mitochondria, they are first processed by other organelles called lysosomes. These contain chemicals called enzymes that digest small food particles that have passed into the cell. Lysosomes are also the garbage disposal units of the cell. They break down worn-out cell parts and gather waste products from the cell's cytoplasm. Another kind of organelle, the Golgi bodies, are the cell's packaging centers. Here, proteins are mixed with other chemicals and enclosed in bubble-like containers that then break away and move to the cell membrane, where their contents are released from the cell.
So that was a quick little video summary of what cells do. One of the aims of this learning unit is to see if you can actually identify the different parts in the cell. So you might want to review that video. The address is obviously on the last page. But here's just a starting example. So if we look at the nucleus in a cell, in this diagram for example, and we can also look inside the nucleus, see chromosomes. I've got some big red arrows showing the nuclear membrane. Remember, membranes are very important in animal cells. So outside of the cell boundary is a membrane. The boundary between the nucleus and the cytoplasm is a membrane. And also, interestingly, the ribosomes are stuck onto membranes in the endoplasmic reticulum. So the endoplasmic reticulum is made out of membrane. Mitochondria are made out of membrane. So membranes are really important. I think that was featured in the video. You could complete this exercise at the end of this unit, and I've got a diagram plus this list in your lab handout anyway. Here's some questions that you might think about, and people have used these questions for writing assignments. What are cells and how are they organized to make animal bodies? How are bacteria different from animal cells? How do bacteria infect animals or their caregivers? Or other types of organisms also, such as fungi, protozoa and worms. So they're all basically infectious. Fungi, protozoa and worms can also infect animals and their caregivers. We'll talk about that a bit later. I want to think about the organization and the hierarchy of animal bodies. And we start with cells, which we just looked at, the model of the cell. Lovely little video. You put cells together to make tissues. And in the example on the right here, we've got muscle cells being put together to make muscle tissue. And I can suggest also that muscle tissue has some connective tissue associated with it. That's the tendons. We also have blood vessels associated with it. So the muscle has blood supply as well as tendons to hold it onto the bones. That then becomes an organ. So we look at a skeletal muscle. It's an organ. It includes the actual muscle cells. It includes some tendon, which is a type of connective tissue. It includes blood vessels, which are another type of tissue. We put these together to make organ systems. So for example, the circulatory system is an organ system, the heart being an organ within it, and the blood vessels are also organs within it. And you put a whole lot of organ systems together to get a whole animal. So the pelican here is built from a number of different organ systems that all work together, like the digestive system, the skeletal muscular system, the cardiovascular or circulatory system, and so forth. And we're going to study the systems, some of them, that function in your companion animals. So we think about cell structure at the beginning. It's the basic unit of structure and function in animals. And virtually all organisms consist of cells and the products of those cells. Cell theory says that the cell is a building block of living things. A cell is derived from other cells, so they divide to make new cells. And the cells contain the information that are used for growth, development, and their functioning. And a cell is the functioning unit of life, so the chemical reactions needed take place in cells. For example, we release energy in muscle cells and convert that to movement. Or in the digestive system, some of the cells make the enzymes that break food down. So the cells do the work. We put them together in organizations called organs and organ systems. Cells are very small and we need to magnify them with a microscope to see them. We use a unit of measurement called micrometers to actually measure cells, and it's usually called microns in slang or jargon. 1 micrometer or micron is 1,000th of a millimeter. And just think about that. A millimeter is 1,000th of a meter. So a micrometer is 1,000,000th of a meter. Micro means 1,000,000th. So cells sit in the size range of about 5 to 500 micrometers, but most of them are somewhere around 10 to 150 micrometers, which is too small to be clearly seen distinctly with the naked eye. Eukaryotic cells, which are most living things except bacteria, such as our companion animals and also plants, protozoa, which are single-celled mostly, and fungi. Protozoa are single-celled animals, and fungi can also be either single or multi-celled. They're all eukaryotes, so they can be quite different cells. And the size of these cells ranging from 30 to 150 micrometers. Cells contain the cell membrane, the nucleus and nuclear membrane, and a cytoplasm. So the cell membrane is the outer layer, the nucleus and nuclear membrane contain the information center, and the cytoplasm is filled with membrane-bound organelles. These are the three major components of the cell. Very small cells, such as bacteria, are much simpler. So bacteria are different from the cells in your normal animal. These are the smallest of cells, and they can be from half a micron to 1 micron in dimension, or easily up to 10 or 15 microns as well. So they tend to be much smaller than eukaryotes. They have a cell membrane, but no nucleus and no nuclear membrane. They've just got a loosely coiled ring of DNA formed into a circular chromosome in their cytoplasm. And there are no membrane-bound organelles in the cytoplasm of prokaryotes. They carry out biochemical functions a bit differently to companion animals. Here's a little bit of a scale of how big things are. So if we think about atoms, we really can't see those unless we use an electron microscope. Molecules are things like water molecules, also protein and fat molecules that build cells, and they get bigger up to the 20 nanometer. And then we start to see organelles, which are built out of proteins, and they sit around about the 200 nanometer range. They can be seen in a light microscope situation. So that's about the limit of light microscopy, which we'll have microscopes in the lab of that kind. Actual cells, somewhere from about half a micron up to maybe half a millimeter or 500 microns. Most of them tend to sit in the sort of 10 or 20 up to 150 microns. It's a wide range of sizes though, and that's not the absolute rule. For example, if you look at a nerve cell, some of the longest nerve cells are measured in centimeters because, for example, they travel from your backbone to your thigh and down into your calf. That's the sciatic nerve. It's quite a long one. Here's just a quick comparison between prokaryotic cells and eukaryotic cells. So the prokaryotes are your bacteria, and the eukaryotes include things like yeast, protozoa, plants and animals, and of course fungi. Yeast is a type of fungi. If we look at the prokaryotic cell, if you look in here, we've got a cytoplasm with ribosomes floating around in it. Now the ribosomes make the protein. That's actually the thing on the endoplasmic reticulum in that video that makes the protein. Here's the bacterial DNA, and it's loosely coiled in the center. It's a big circular molecule. It's actually called a chromosome. We've got a cell membrane in this bacterium and also quite a thick cell wall. Now some eukaryotic cells have a cell wall. We're thinking fungi and plants primarily. Alright, animal cells don't have a cell wall. We just go in and we have a membrane, and then we have mitochondria and other organelles inside the cytoplasm. Here's the Golgi body. Think back to that video, review it. We also have ribosomes in the cytoplasm and attached to the endoplasmic reticulum. And here's the DNA inside the nucleus, which is separated from the rest of the cytoplasm. Quite a different layout to the bacterial cell. Let's think about the bacterial cell, or prokaryote. We call them prokaryotes because that's the technical name. They're small and tend to be single cells or unicellular. The bacterial cell wall, often called a matrix, is outside the membrane. It doesn't have a true nucleus, just a circular DNA molecule, which is considered to be a single chromosome. And you may have smaller loops of DNA, which are called plasmids, sitting in the bacterium as well. They don't have defined organelles. That's a feature of them. So they lack defined organelles. They can be motile, so they could have flagella or cilia. And examples of these include the archaea, bacteria, and cyanobacteria. So archaea sit in the middle between bacteria and eukaryotes. Let's run down through a eukaryotic cell. So some of the features here, most importantly, they have a true nucleus. They have one or more chromosomes, which are linear or straight molecules. They're not circular. They have membrane-bound organelles inside the cytoplasm. So we'll see the Golgi, which is, remember, the packaging apparatus, the mitochondria, which is the power plant. In a plant, you'll see chloroplasts, obviously not in animals. Chloroplasts generate energy. We also see vacuoles, such as the lysosome, which is managing food coming in or managing garbage going out. The endoplasmic reticulum, which is sort of represented by these little membrane slabs here, quite important as a processing factory for proteins. In plants and fungi, there's a cell wall. In animals, there's just a membrane. And they can also move, so many plants can disperse using seeds, floating seeds, or pollens. Animals tend to move, and small animals may move by flagella or cilia. Large animals, like your companion animals, tend to walk along, and we'll deal with that as we move through. Eukaryotes can be single-celled organisms, such as protozoa, or they can be complex multicellular things like plants and animals, and that's really where we dwell on most of the time with our companion animals. I'm just going to run through some shapes of bacteria. So we see cocci, which are circular or spherical. When you look at them down a microscope, they look like circles, but they're actually three-dimensional, so they're spherical. We also have bacilli, or rods, that are cylindrical, and we'll hopefully see some of these in the lab. We don't want to see curved molecules because they're often curved bacteria or helical bacteria because they tend to be a little bit more nasty, such as Leptospira, which is a curvy spirochete, and that might be what it looks like there. They tend to be a little bit nastier. The simplest ones may be just a curved arrangement. Here's an overview of bacteria, and they can vary quite a bit in shape, but you can see in this one the flagella. We also have bacterial pili, which in some cases are used by bacteria to join with the next bacteria over in the broth, and they can share DNA through their pili, and they swap information such as antibiotic resistance. That's something to remember. And ribosomes, and we've got the membrane here, and we've got this outer layer, which is the cell wall. So that's just a little review of the bacteria. And I want to go quickly over the bacteria. Sometimes outside the bacteria we see a capsule or a slime layer, and that helps the bacteria stick. They can also stick to surfaces and grow this way. They have the cell wall, which is a complex structure. It's quite rigid and it gives them some shape. And it's located outside the plasma membrane. And it also protects them from environmental damage, gives them a little bit of resilience in the environment, which means they can last between one animal and the next when they're being passed on. They have a plasma membrane which allows things to pass through, such as oxygen in and wastes out. So we call that semi-permeable. And here's a sort of diagrammatic view of it all. I'm not going to dwell here. You can have a review of this PowerPoint, which is in your Moodle anyway. Bacteria grow very quickly and they can multiply very rapidly, although sometimes can be slow as well. So depending on the bacterium. E. coli, look, takes 20 minutes to copy itself out and divide. After several hours, you'll have 1 million or so E. coli sitting in a little colony, and when they grow enough of them, you can see them as a little circle or a dot or something like that on a agar plate, and you'll see those in the lab. Very slow-growing bacteria include things like mycobacteria, which may be an issue for some farm animals, such as cattle. Bacteria tend to be grown on culture media, and you'll see this in the lab. We'll mainly be using plate media, such as what you see at the top, but we'll be using a little bit of broth for some of our experiments as well. We can have defined, which is synthetic with the chemical composition known, or complex media, which might be a bit less defined, such as blood medium or yeast medium or serum containing medium that contain a few more growth factors. And these sorts of bacterial media cultivate quite a large range of bacteria. And here's an example of blood media, and you might see that in the lab, and also nutrient agar, semi-solid and defined. So the blood agar is complex and has all sorts of stuff in it, and things can also dissolve it, which you will see. Here's someone streaking a plate, and this technique is called a streak. We're going to do this quadrant streak, which is shown diagrammatically, just so you can get the idea. We can also grow bacteria as a stab in a little tube of media, or as a lawn, and in a range of different ways. Here's a bacterial growth curve, and this is probably based on something like E. coli, which is fast growing. It starts out slowly. The bacteria take an hour or two to get growing in the media before they start multiplying rapidly. They'll grow through what we call the log or exponential growth phase very quickly until they reach a maximum for the for the media they're on. So once the media is saturated with bacteria, they'll stop growing and you'll tend to get a die-off. Depending, sometimes they can go back up here too. Sometimes we'll see this growth curve go back up, but that's the bacterial growth curve. Generally, if you exhaust the media, the bacteria are going to tend to die until you put them into fresh media. That's only relevant when you're culturing them, because if they're inside an animal's body, they're going to have constant access to fresh cellular material upon which they can cause havoc. We'll look at this pop quiz when we get into the class, but think about these questions because we'll discuss these in class. What do bacteria need to grow? Can they survive in the environment, and how do you think that works? And how do you think you can stop bacteria in the environment, for example, of your animal facility? Think on those things because we'll do that in the lab and discuss it as well. I want to talk briefly about different types of eukaryotic cells, and you can see up in the right hand corner a single-celled animal, which is probably a plasmodium or something like that. Sorry, not a plasmodium, it just looks like a nice ciliate. But anyway, single cells of eukaryotes. On the left we can see the egg or ovum, which is the first cell before fertilization of your animal. And over here on the right we've got a nerve cell. Now remember, nerves could be quite long. If this nerve cell's going from your spine all the way to your calf muscle, it could be quite long. It could be several centimeters long, or 30 centimeters or 40 centimeters, what have you. That's the axon, which is the extended component of the nerve. This is an exception to the general size of cells because they do get quite long. And then we have the white blood cell, which is a leukocyte. Don't forget there's red blood cells as well, but these are the ones with a nucleus in blood. And we have a sperm cell, which is the other half of the ovum. So they're both called gametes. When you combine sperm and ovum together, you begin a new life cycle. I'm just going to go through the parts of an animal cell. And if we look at this, so we've got the cell membrane, it's the external boundary, and it's shown here as this black line. The nucleus is the control center. It's got the green nuclear envelope, bright green here, and it's shown as a sort of a browny color on the inside. That's where the genetic material sits. And if you look at the picture on the right here, this blue colored stuff is the cytoplasm. And inside the cytoplasm, or the cytosol, as it's called in some books, you'll find the rest of the organelles. So we've got the lysosome sitting up here, which is, remember, the garbage system, digesting unused material or digesting food perhaps to make it available. We've got vesicles of different types, Golgi apparatus with little bubbles coming off the Golgi that might move to the outside of the cell and be secreted. Peroxisome, mitochondrion, endoplasmic reticulum, and that's rough and smooth endoplasmic reticulum. The lysosomes are the digestive part of the cell, and they're responsible to break down, for example, they can break down bone and release calcium. They're that good. They can also break down waste products in the cell. The mitochondria are the powerhouse, remember that one? And the ribosomes, which you'll find sitting on the endoplasmic reticulum or free in the cytoplasm, synthesize proteins. So, endoplasmic reticulum has, the rough endoplasmic reticulum has ribosomes to make protein, and the smooth endoplasmic reticulum, which doesn't have ribosomes compared to the rough endoplasmic reticulum, is involved in making fatty acids or synthesizing fatty acids. The Golgi transports these proteins and can move them to the outside of the cell. So for example, if you're making a digestive enzyme, you package it in the Golgi apparatus and you export it to the membrane where it's released. In this example, it might be released into the duct of the pancreas or something like that. And then it can help with the digestion by entering into the intestines. And the centrioles are very important for the division of the cell. Now, not everything's shown in this model, but if you have a look at the other picture that we've got, it's all there. And here's a more detailed model. The picture that we use in the lab's also got most of these things on it. There's actually quite a few beautiful pictures you can get out there. We're going to go through this in your groups. When we're in class, we might talk about organelles, what they are, what are the components in a eukaryotic cell, so you can prepare for this by thinking about this. Are any present in bacteria? Which? If any? How many types of animal cells can you brainstorm as a group? That would be an aim when we get to the lesson. Organisms can be quite complex. Organisms are the animals and so forth. And just a quick reboot of our cell theory here. Complex multicellular life needs to grow, reproduce, to live and to die, and it's done through cells. So the cell theory reboot, we start with cells. They're assembled to make tissues. They're then assembled to make organs, and those organs make organ systems, which turn into the whole animal when they're all put together. The whole system can be broken down or built up in this way. I'm just going to play this video. I'm going to escape from this PowerPoint momentarily. You can watch this at home. There's the address down below. So this is a very cute little development of the zebrafish embryo. It's a very short video. This is the first cell that's formed when a zebrafish egg is fertilized. All the cells on top are dividing madly, and it's about to change shape quite dramatically. So this big ball of cells on top is now going to squeeze itself around the yolk in the middle. So that yolk is going to stay there for a while and help feed the developing zebrafish. As we go through to about 24 hours development, you can see that we've got the head at the top there. I'm just going to reboot it. We'll watch it again. So these cells are dividing madly, forming a ball of cells. And when we get past this shape change, you'll see the head at the top, the tail at the bottom, and all the blocks of muscle in between. It's a lovely little video watching this divide, watching the cells divide and form the new fish. So at the top we've got the head. You can start seeing the brain and eye developing, the tail to the bottom, and the yolk's still in the middle. So the yolk's quite important. The zebrafish is going to grow and develop in its early days using the yolk. All life starts as a single cell, so we think about the sperm and the ovum combining to make that first cell. It forms a ball of cells, slightly different in a mammal like the elephant compared to the fish. These cells divide up and make more cells, and then the fetus starts to develop by interaction with these cells. So we've got here a 2-month-old elephant fetus. It's quite well developed at this stage. We can already see its eyes starting to form. They're not yet fully developed. We're at about that point. Here's just some credits for this video. I'm going to shut down in a moment. Thank you for watching this, and I suggest that we'll think about these questions that are embedded in this particular video when we get to your block course.